Hi everyone, you are watching Biology with Intra. So, I just want to wish you guys a warm welcome back to my Facebook page or YouTube channel, and I want to start by saying that I've decided to do something very interesting and something very tremendously awesome, and that is that I've started making videos on AS level biology, which is 9700. Now before then I was making videos on IGCSE and O levels biology, and there's not much difference between O levels and IGCSE. Anyhow, I still made videos on both of them, and I was also making past paper questions on O levels biology on both paper 1 and paper 2. And there's two major announcements. With the first one being that I'm making AS level biology videos 9700, and in front of you is a video that I'm going to upload on the very first chapter of AS biology, right? And I'm also going to start making past paper question videos on IGCSE biology, right, on both paper 1 and paper 2. So there is some difference in the question paper of both IGCSE and O levels. Nonetheless, I'll be making the videos on all those question papers, and I'll cover the techniques and the tips and all those golden things you'd want from a teacher, and that will all come from me free of cost. So do share this around with your friends and families. Spread the word, because I'm spreading free and quality education, and I really hope you guys like this. So do enjoy this video for me. Now before we start, I just want to give you a brief introduction about AS biology. So in AS biology, you'll be giving 3 papers. That is paper 1, paper 2, and paper 3, right? So paper 1 is your old and regular MCQs. Paper 2 is your structured questions, just like your O levels in IGCSE, and your paper 3, which is something a bit new, is where you actually have to perform your practicals. Yes, you have to perform it physically, and the good thing about that is that you'll be given a set of instructions on the question paper, so you don't have to memorize anything. You just have to follow what the examiner tells you to, and just do it the right way, and you'll be judged based on your performance and on your method of recording your results and the output, right? It's not that hard. It's very easy. It doesn't take a long time to prepare for that, because our main focus is paper 1 and paper 2. Nonetheless, paper 3 is of importance. Don't get me wrong. Paper 3 is actually very important, and if situation and time permits, I may make videos on paper 3 and as to how you can perform paper 3 by performing some experiments at home with some of my very own apparatus, chemicals, and etc. Okay, so let's get started. Now, the first chapter of AS biology is again cells. So just remember, whenever you start biology, let it be O levels or A levels or anything outside of this, always expect to start with cells, right? Now there's some difference. There's a lot of difference between the O levels and IGCSE cells and the AS biology cells. This one is a bit more detailed, a bit more complex, a bit more lengthy. Plus we have this new area here. This new area is on your microscopy. Now I'm not we did not have microscopy in O levels biology nor IGCSE, although we had some magnification calculations in paper 6 in the ATPs of both the papers in O levels and IGCSEs. But here you need to study a bit more about microscopes, gets into a bit more detail, but don't worry, I'll be covering all of that in great detail. Okay. Right, so let's start by working on some of the basics that you need to know, right? So what are organelles? Now we have discussed about organelles in O levels and IGCSEs, but let's do a bit of a recap, but in a different manner, right? Because you're in an advanced level now. It's not your O levels or IGCSEs anymore. So organelles are a functionally and structurally distinct part of a cell, right? So what this means is that it is like an organ, right? Just consider it to be like an organ that structurally is unique and functionally is unique. So what that means is that it has its own different structure and it has its own different function as well. Like if you take the nucleus, structurally it is spherical and functionally it controls the cell, and that is unique. And you should expect the function and structure to be different for many other parts of a cell. Get it? So this is what the definition organelle means. Organelles work in their separate compartments, which is separated by a membrane, and this is known as compartmentalization. So what this word here, this big fancy word here, compartmentalization, means is that each and every individual organelle has its very own function, and it separates in its very own compartment, which is separated by a membrane, right? And this whole thing is known as compartmentalization. Let me demonstrate by drawing it up for you guys. So let's consider that this here is an organelle. Sorry, a cell. Yeah. So let's consider this to be a cell. This is one compartment that's circled and green. This is the second compartment, and this is the third compartment. In this area, you may find the maybe the nucleus or the endoplasmic reticulum, and maybe here you may find the Golgi body. Here you may find the lysosomes. So basically, they have their own membrane. This line you see here is it's a membrane that separates the function of the organelle with that of the cytoplasm. They need to be separated in order to happen properly, right? Like, for example, let's take a bit more closer look. Let's say this is a nucleus, right? And then this is a cytoplasm around it, right? So the nucleus has its membrane so that its functions do not mix up with that of the cytoplasm. And this is done with the help of the membrane of the nucleus, right? This is needed. This is very important. Or, you know, the functions of the nucleus would mix up with that of the cytoplasm. So they need to be kept separate. And so you can consider them to be maybe like a compartment, right? So this is the nucleus, this is the compartment of your nucleus, this is the compartment maybe of your nucleus again, this is the compartment of your Golgi body, this is the compartment of your lysosomes, right? This is what that meant. So the sharing of work between the different organelles is known as division of labor. Here's the thing. So let's take an example. You're in your house, so your mom or your dad or whoever cooks in the kitchen, and then there's someone you know, cleaning the floor, and then there's someone who gives you the money, and then there's someone who ensures that your pipes are working fine. So basically, the person ensuring that your pipes are working fine would be an electrician, or maybe a plumber, or whatever. The person ensure that you get money, like for your education and stuff, is your dad, right? And for stuff like to get food and to be taken care of when you're sick and to get some love and stuff, that's from your mother, right? So this is a very typical stereotypical example, I know, but let's keep it for the sake of understanding. So what this means is that this basically means is that you know, they are all working in their compartments. Like, to get your food ready, your mom has to work in the kitchen. That is her compartment. To get money, your dad has to work in the office or somewhere outside. That is his compartment. And to get your pipes working, the electrician has to work in the bathroom. That is his compartment, right? So they're all needed for a fully functioning house. You cannot expect a house to function without money, without food, without love and stuff, right? So similarly, the organelles in a cell basically work together. You know, division of labor means they share functions. They help each other out to ensure that one particular function is carried out. For example, for the synthesis of ribosomes and for the production of ribosomes, the ribosomes and I think I said it the other way around. What I meant was that the synthesis of proteins, I'm very sorry about that, the synthesis of proteins and for the delivery of proteins out of the cell is done with the help of the ribosomes, the Golgi body, the rough endoplasmic reticulum. So they all work together for a common purpose, and that is called division of labor, right? Sharing work. So to remember that, right, it's very simple, not that hard. Okay, let's move on. So, by the way, we'll be talking about the Golgi body and the ribosomes that I just spoke about there, so don't worry about that. I'll clear everything out. Below are some of the organelles that are found in both an animal cell and a plant cell. So here's how we are going to cover this chapter. We're first going to talk about the organelles found in a cell, but before we do that, let me just explain the sub part of that. We're going to talk about the organelles found in both an animal and a plant cell, right? In both the cells. And then we'll talk about the organelles found only in a plant cell. And once we're done with the organelles part, we're going to come to microscopy. And basically, it's not that hard. It involves some calculations, some conversions. And, you know, there are two types of microscopes, light microscope and your electron microscope. So I'll cover that up. Don't worry about that, okay? So here, just take a quick glance at this list, right? These are the organelles found in both an animal and a plant cell, excluding the ones only found in a plant cell. The nucleus, cell membrane, cytoplasm, you know, you can just have a look at it, right? You may come across some new names here. Don't worry about that. Like I said, this is a bit more detailed and complex, but not that hard, okay? At the end of the day, it's not that hard. It's going to be a piece of cake. Okay, so in front of you is a picture of a typical generalized animal cell in which the labeling has been done very clearly, and I hope the picture is also very clear to you, in which the organelles that we're going to discuss have been labeled here. So we're going to have a look at them one by one, and we'll start with the nucleus first. So let's have a look at the nucleus first. Okay. So this is the nucleus, right? And if you come here following the arrow, this whole thing here, I'm going to grab a pen to kind of underline that for you guys. This whole thing here is a nucleus. It's at the middle, it's big, it's deeply staining, due to which it can be easily seen under a microscope. These are some points, do note them down. Okay, and there are even more things present in the nucleus, like this is the nuclear membrane here, this membrane here that you can see, right? Also called the nuclear envelope, right? And then there's some more parts which are not labeled here, but we're going to have a look at them in the next slide. Okay, so now you can see that there is your bit more detailed structure of the nucleus, in which let's have a look at it one by one. So this here is your nucleolus. Now the nucleolus is present inside the nucleus, right? It's also somewhere at the center. It's present inside the nucleus, so don't confuse this for the nucleus. This is actually in the nucleus. This whole thing here that I'm kind of moving my cursor around is the nucleus, right? I'm just going to draw a line to show you that this whole thing is the nucleus, and there are some things present inside the nucleus, right? So there are some pores on the walls of the nucleus called the nuclear pore. These are just some holes that you can see here, here, and here, right? And there is this liquid in the nucleus, the nucleoplasm. It's a dense liquid. And then there's your genetic information, chromosomes or chromatin. Now we won't get into the difference between chromosomes and chromatin. That's to be done in another chapter, right? But just know that during cell division, your chromatin becomes chromosomes, right? Deeply coiled. But I'll cover that in one of the future videos. Don't worry about that. Here we go. This is the nuclear envelope. The nuclear envelope is actually the membrane of the nucleus. This whole membrane, just like a cell has a membrane, the cell surface membrane, this is actually a very own membrane of the nucleus. Remember I spoke about divisional labor and compartmentalization? Like I said, every organelle has its own envelope, right? Also do remember that the nuclear envelope, is continuous with the rough endoplasmic reticulum, right? You can see here, right? This is your endoplasmic reticulum. These compartments, not compartments, but like structures maybe in a sort of pattern here. You can see that they are continuous with the nuclear membrane of the nucleus. The nucleus has two membranes. That basically means it's called an envelope, right? I'm going to show you what I mean by that. Going back to this slide, you can see here that the nucleus is indeed continuous with the nuclear envelope, is indeed continuous with the rough endoplasmic reticulum right here. So just remember that, and we'll be talking about that in the future. Okay. So the nucleus is the largest organelle. Yep, that's true. It has a double membrane called an envelope. Now, what I mean by this, it's not that much of a rocket science. Like, this is the first membrane of the nucleus, and then there's a second membrane of the nucleus. There isn't much space between the two membranes. My drawing isn't good here, so just get the idea. And this whole thing that I'm shading is your nucleus, okay? So this whole thing here is called an envelope, right? A double membrane. So exchange of substances between the nucleus and the cytoplasm occurs via the nuclear pore. So these holes that you see here, these are the holes through which things can leave or exit the nucleus. So stuff like ribosomes and RNA leave the nucleus for protein synthesis. Now we'll be covering that in one of the future videos. Protein synthesis ribosomes are also an organelle that makes proteins, and RNAs, messenger. There are types of RNA, messenger RNA, transport RNA, and we'll be talking about that in the future. But just know for the sake of this video that ribosomes and RNA leave the nucleus. Materials like ATP, hormones, and proteins enter the nucleus. Hormones enter the nucleus, and so do proteins, because proteins are needed to make ribosomes, right? So basically the proteins enter the nucleus, and the ribosomes are made, and then those same ribosomes exit the nucleus. And ATP, we just have spoken about that. It is actually a currency for energy, or energy carrying molecule known as adenosine triphosphate. Nucleoplasm is a dense liquid that contains the genetic information, called chromosomes or chromatin, that are thread-like structures which are made from a chemical called DNA. Both chromosomes and chromatin are made from a chemical called DNA, aka deoxyribonucleic acid. The nucleus controls, basically the main function of the nucleus is to control the cell activities, right? It acts like the boss of the cell, controls the cell division process, cell activities, giving instructions and all that stuff. Basically the recipe of life is in your nucleus. And the nucleus here, this deeply staining thing here, makes ribosomes, right? This is the part of the cell responsible for making ribosomes. Note that your nucleus is even more deeply staining, and so are your chromatin and your chromosomes, and expect there to be from 1 to 5 nucleus in mammals in particular. You may read the A-levels AS book by Mary Jones to get a better understanding. Hopefully you'll understand it word by word, as I am covering almost everything from there. Now we're going to talk about your cell membrane. Now before we look at the cell membrane, let's first have a look at it from the animal cell first, so that we are clear. Where is the cell? Yeah, this is the plasma membrane, the cell membrane, the outermost membrane of an animal cell, right? Just have a look at this first. Yeah, okay. So I know this is a bit detailed structure, and you don't need to go into the detail of this. You really don't need to go into the detail of this, as we'll already be covering this up in chapter 4. You know what the different types of lipids and proteins that make up your cell membrane, their functions and stuff like that. So just ignore this. Don't spend too much time on this. I'll be making a video on this, but just get the idea. This is how a cell membrane typically looks like, to open up your mind, because this is AS biology now, guys. This is what you'll be studying in chapter 4, right? But you just need to know the cell membrane from this structure that I just showed you, right? This is your cell membrane. There we go, right? Cell membrane. That's it. For that, leave that picture for now. I just post it here for the sake of information. So, so it is also called a plasma or a cell surface membrane. It has about 2 more names, so just keep that in mind, okay? It is semi-selectively or partially permeable. This means it only allows some or certain substances to get into and out of a cell. It basically is very choosy when as to deciding what enters or leaves the cell. Controls the entry and exit of materials. This is the main function here that I'm going to highlight of the cell membrane. This is actually, well, it must be easy. Controls the entry and exit of materials, right? And this is a bit more detailed part coming. And the cell membrane is composed of lipids and proteins, and there's some examples of lipids like phospholipids. We'll be talking more about phospholipids in the 2nd chapter, which is your biological molecules, the structure and everything, okay? And some protein channels. We'll also be discussing about that in chapter 4. Now, it's very thin, and it has about 3 layers that are called the trilaminar layers, okay? What I mean by this? What I mean by this is that the cell surface membrane is made up of 3 layers, right? This is the first one, the second one, the third one. This is the outermost layer, this is the innermost layer, and this is in the middle. They're very thin. There isn't much space in between them, but I've just, made it roughly. So basically, this whole thing is called the trilaminar layer. Remember that. Now, let's talk about the cytoplasm. There isn't much about the cytoplasm, but let's still discuss it. Cytoplasm is a jelly-like substance that is in aqueous water solution. It is the area where all the metabolic reactions occur that are controlled by the enzymes. So this cytoplasm is basically where all the chemical reactions occur, where all the organelles are held in a fixed position due to the jelly-like consistency, and they contain some mineral salts, some ions, some sugar, right? So basically this region between the nucleus and the cell membrane is your cytoplasm, right? I'm just gonna shade it up for you guys with some lightly colored pen to give you a general idea. Like, consider this to be, this is the cytoplasm that I'm coloring, right? In which all the organelles are held or in a fixed position, right? Don't consider these dots to be the cytoplasm. They're basically your ribosomes, right? Or maybe lysosomes. We'll get to that, okay? I'm gonna circle that up for you guys so that I don't mistake them for organelles. This, and then these stuff, and then these stuff. Remember, they're not the cytoplasm, they're organelles. They're very small, so it's a bit difficult to identify them, so just remember that, okay? Okay, so where were we? Yeah, so we're done with cytoplasm. Like I said, there isn't much to do. So let's talk about a brand new organelle that it may be your first time studying, I'm not sure. Lysosomes are structures in which digestion of unwanted parts or even whole cells occur. they contain hydrolytic enzymes that get rid of any foreign objects like bacteria or other germs. The thing to understand here is that your lysosomes are structures that contain enzymes, and when something bad or something dead is in the cell, it basically takes it in, absorbs it in, eats it in, or engulfs it, and the enzymes within the lysosomes break it down so that they are of no danger to the cell, right? When I say hydrolytic enzymes, what I mean by that is that they're acidic, right? Any foreign objects like maybe bacteria, germs, or anything that may cause harm, or something that's dead, or something useless, or it's not needed, is actually engulfed by the lysosome and broken down with the help of the enzymes. The enzymes play a big role here in the breakdown, right? This is a typical structure. It also has a very own membrane. It has a single membrane, and these are some of the enzymes that you can see here. And then foreign objects like this would be entered. Basically, this lysosome here engulfs it, and upon engulfing, the enzymes do their work and they are destroyed, right? They're destroyed, so let's consider that I'm destroying them right now. So when they're destroyed, they're gone. This is what happens in lysosomes. Basically, I'm just telling you this as extra knowledge. They're also called suicide bombers, something like that, because what happens if all the enzymes from the lysosome get leaked in the cell? If that were to happen, then those enzymes would be killing other useful organelles that are tremendously needed in a cell, right? Like if the enzymes in the lysosomes make their way out, they'd be killing the nucleus, the cytoplasm, and some other parts of the cell membrane, and that would be deadly, right? Okay, so I don't think the lysosomes are here in the structure. This is the mitochondria, ribosomes. Sorry, lysosomes, not ribosomes. Nope, they're not here. But nonetheless, just know that this is what your typical lysosomes look like. They're not that big, not that small. Here's what I'm gonna do. Just expect them to be something like this. You know, like circular, like this much size. This is their size. It's not too big, not too small, right? Okay. Now we have another thing to cover, and that is your microvilli, right? So your microvilli is not shown in the picture above, the one that I just showed you, a couple of seconds ago. So I have this new picture for you that should hopefully clear it out of what microvilli are. So this is your microvilli, and they are found on the cell surface membrane, right? They're found on the cell surface membrane. Here we go, microvilli. This is the cell surface membrane, and on the cell surface membrane, you can find the microvilli. So the microvilli are finger-like extensions that increase the surface area to volume ratio for absorption. They can be found on the cell surface membrane. Basically, the cell membrane is absorbing stuff. And to make that happen faster and efficiently, these microvilli are long and thin. They increase the surface area to volume ratio to increase the rate of absorption. And note that they're also found in the small intestine in the villi. Remember, microvilli, that they increase the surface area to enhance absorption. And this is what your typical microvilli should look like. They protrude from cell membrane or from maybe an epithelial cell, and they are long and thin, and they're moving about like this in a wave-like motion to increase the surface area to volume ratio, right? Okay, guys, that is it for today. This is a very big topic, and it is not possible for me to cover it in one go, so I'll be dividing it up. So hopefully in the next video we should be done by organelles, like completely, right? Mitochondria is a bit detailed, chloroplast, which is again found in plants only, they're quite detailed, right? There's a lot to understand about them, and even more about the rough endoplasmic reticulum and the Golgi body and the smooth ER. So just wait it out, and I'll make a video on that very soon. And once we're done on your cell organelles, we'll start out with microscopy, and I'll try to finish that off real fast, real quick, so that we can move on to some past paper question videos. And I'll be taking and continuing O-levels and IGCSE videos parallel to AS biology, so don't worry about that, okay? And yes, we will also be we are actually working on A2 right now, so we'll be making more videos on, sorry, not more, we haven't made any yet, but we will be making videos on A2 biology, so be ready for that. There's a lot to come ahead of you, so be prepared. So I hope you enjoyed this video. Have a great day. Do share this around, spread the word, subscribe, like my YouTube channel, comment below, like and follow my Facebook page, react to this video, share this around, and I really hope to see you guys share this around. And the more we grow, the more the beneficial it is to you guys, for the biology learners out there. Well, that's it for today. See you next week.